It's my pleasure today to introduce Dr. Daniel Allen. Dan is an associate professor in the Department of Ecosystem Science and Management at Penn State, where his lab focuses on two main areas of research, the macrosystems ecology of stream drying and global change in freshwaters using data synthesis. Today's talk towards understanding how drying impacts streams across biological and spatial scales, I think, is of particular interest, especially to those of us in the West this year, since we've had such a dry winter. So thanks for being here today, Dan, and take it away.
Thanks for having me. I'm really excited to be here. Everyone can hear me, I hope. If not, just ping me. I'll keep going as if everyone can hear me well. All right, so the Earth is getting drier. This is something that we know. It's all over, all the continents, all over the Earth, North America, South America, Asia, Africa. On every continent, we're seeing lands that have a drier climate. And this is particularly true in Europe. 96% of land is drier than it was. North America, it's 69%. And then this is something that started to happen in the 1990s, and it's something that is continuing. We've had some record weather events recently. You might remember 2022 was one of the driest years on record in California. Lake Mead, much of it dried up. In 2024, even the East United States, which we typically think of as being wet, we had wildfires. That was news to me here in Pennsylvania. I didn't really know that we had much wildfires, but we actually did have quite a few wildfires that year. And then, of course, as was mentioned, right now there's huge record low snowpack in the Western United States. This is a picture of the California Department of Water Resources crew going out to measure snow on April 1st in the Sierra Nevadas, and they're having a little trouble. And you can see on the SNOTEL map on the right, which I just pulled this morning, shows just how low the percentiles are for snowpack across the Western United States. So it's going to be a very dry summer, and we aren't really looking forward to that in the Western US. So I'm going to start just by sort of giving a brief outline of the talk. First, I'm going to talk about how streams dry and how that is changing. Then I'm going to talk about some results from a big collaborative project that we did on looking at how stream drying affects stream invertebrates. I'm also going to talk some about current and future projects. And so I'm going to start just by thinking of our definition of a stream. And so a stream is a body of running water, such as a creek flowing on the earth. That's from the dictionary, right? And so if you think about a stream, picture that in your mind, you're going to picture flowing water. That's what we think a stream is. It's the definition. But what we know is that most of the rivers on Earth do not dry. More than 50% of them do not dry. And again, these are widespread across the globe on every continent. And this is a map from a paper that came out in Nature a few years ago that showed this. The United States as well, 60% of the streams in continental United States dry. This is a map here showing the different watersheds with the total percentage of dry stream length. And we see, of course, there's more dry streams in the West than there are in the East, but still here we still have a lot of drying even in the Eastern United States. And so for this first part of the talk, I'm going to talk about a lot of work that we did as part of the Dry Rivers Research Coordination Network. And this was a network funded by NSF and RCN, where we had about over 50 participants from over 3 continents. We have ecologists and hydrologists. And so we sort of all got together to try and study how we study streams. And from ecology and hydrological disciplines, and how we might be able to advance our knowledge of drying streams. And so what was predicated by this is streams for a long time were largely ignored by scientists. And this is one of our first papers. And this is both from hydrological and ecological perspectives. These streams were just not really given much attention by researchers. And this is actually a bit of a problem because we now know that they are so pervasive and they are increasing. And for ecologists, this was a paper that I led where we reviewed all of the different stream ecosystem conceptual models that had been published to date. And we sort of reviewed all these conceptual models to think about if these stream conceptual models were relevant to drying streams. And only a couple were directly relevant, several were indirectly relevant. And so even from an ecological perspective, we really don't have much knowledge about how these streams should work. And then for hydrology, we also looked at how we study dry streams, how we're getting data on dry streams. And so we looked at the global distribution of stream gauges to assess sources of bias in stream gauge placement all across the globe. And we used a method where we looked at a variety of variables to determine which variable produced the most bias in terms of affecting the network as a whole. And what we found was that human footprint was the most important driver determining whether or not a gauge was placed in a stream. And that makes sense because, we're humans and we put water monitors out where we are so we can monitor water levels because we're interested in them. Because they're a resource for us to use and manage. But flow permanence was the second most important factor influencing stream hydrology gauge placement. And so really it's even hard to study these streams from a hydrological perspective because they aren't gauged like perennial streams are. And so the next thing we wanted to do was to sort of see which gauges actually had data for dry streams, and so we could study how they dry and how that might be changing. And so for this part, I'm going to talk about two papers that we wrote as the RCN, one looking at how drying patterns varied over space, and the second looking at how they varied over time. And so here is a map showing our site selection. So we used the United States Geological Survey stream gauges for this study. And what we found here were we did find 540 gauges that showed drying at some point in their record. And so we classified these as streams that only had 10 years of data, had somewhere between 5 to 360 days of no flow. And so they're either, some of these were sort of, quote, reference gauges in pristine conditions, and some were non-reference gauges where the flow is heavily impacted by humans. And so one of the things we had to do was figure out how we wanted to classify no flow metrics hydrologically, because there aren't that many flow metrics out there for them. And so if this is our average stream graph here, we defined this dry period as the annual duration of no flow days. That was our first variable. The second we looked at was the rate, which is sort of the number of days it would take from a peak flow to go down. Let's see, can you see this? From down to the bottom. And then the other aspect that we looked at was the timing. So which time of year did we have the first no flow day? And so the first thing we looked at was the spatial patterning, how these varied across the United States. And so our no flow duration was generally greater in the South and in the West. And here I'm going to show several graphs that look like these. Here are our gauges. We have the reference and non-reference in circle and triangle, and then the color scale representing the data. Here, this is the no flow fraction. Being very wet is dark, and being very dry is lighter in color. For the first date of no flow, we also found that date occurred more typically earlier in the year in the Western United States than in the East, and then also in the North a little bit. And then when we looked at sort of that drying rate, that peak to no flow, there we found less pronounced spatial patterning. And so this is just sort of some data just sort of showing, sort of confirming what we generally expected, that when we have more arid climates, such as in the West and in the South, we're going to have more drying, and that drying might happen earlier in the year. Now, the next thing I wanted to do is I wanted to look at how these were changing over time. And so these graphs are the same set of gauges, but here within each gauge at these three metrics, we looked at whether or not over that time span of at least 10 years for each gauge, whether or not that trended wetter or trended drier. And by drier, we mean less flowing days, and by wet, we mean more flowing days. And so it was interesting here with the no flow fraction was what we found was widespread trends towards drying, especially in the West, but also in the South. And we also interestingly found some trends toward wetting, meaning more flow, particularly up in the Midwest, the northern Midwest. And what we actually think might be driving this is that this is actually streams just being frozen for less time of the year. So there is more flow because it's not zero when it's frozen, which is hydrologically on a gauge reads the same as if it's dry. For the timing, we also found some significant trends where in most cases, when there's a change, it was trending to dry earlier. And again, it's more in the South than in the West. And then for the rate, here we are also seeing changes, and it's a little bit more mixed again, just like it was before, where the spatial patterning is a little bit more scattered. But what was really notable was just how many of these gauges are actually changing over time in the way that they're drawing. Over half of these gauges had a significant trend in at least one of these three metrics, right? So it just means that over this time span that we're studying here, the way streams are drying are changing very, very rapidly. So next, I'm going to talk a little bit about how that might affect stream communities. And so the results of this project are a big collaborative project that we had called Stream Climbs. This was an NSF macrosystems project where we had researchers across 10 different US institutions where we studied how drying impacts populations, communities, and ecosystems by looking at different aspects of the ecosystem. For the population, we had a part on genetic diversity in invertebrates. For the community part of the ecosystem, we're looking at taxonomic diversity. And for ecosystem level responses, we looked at food web structure. So here what we're going to do is just sort of talk a little bit about the overall design and our approach. And so what we had here was basically what we would do is we would go to an individual site, and this in some ways sort of mirrors what NEON does, where we're trying to create a standard set of methods across a wide variety of stream types. And so that was a bit of a challenge to figure out how to do that. But what we're doing a little bit differently than NEON is within which watershed we're nesting this sampling design. So within a given site, we have up to 10 sites within each watershed, and we pick these sites to try and represent the different conditions that exist in the watershed in terms of stream size, but also in drying. And then we also replicated these watersheds across regions. So we would have multiple watersheds in different regions. And then finally, we did this across the southern United States. So we had 10 different basins replicated in different regions. So we had 100 sites total. And so we did a bunch of different things at these sites. But first, what I'm going to talk about first is talk about Albert Ruhi, who is associate professor at UC Berkeley. His lab led a really cool part of the project looking at the diversity of benthic macroinvertebrates. And so one of the things that Albert and Kyle Leathers, who got his PhD on the project, and they led was sort of these questions of sort of thinking about, because we have this nested design within the watershed, we can start to think about what the effects of local drying disturbances are versus these network level fragmentations, these network level effects of drying on local diversity, and then also thinking about how these two mechanisms might interact. And this part of the study, so first what they did is they developed this model, this mathematical model to try and predict these dynamics. And then using our field data that we collected in the field to evaluate how those data compare to model expectations. So first, I would say with this result here, this is if you look at this with this diagram, what this is showing is essentially a stream network. And this is the replication of the stream network, one of the study sites that we used in the project, and it's a mathematical representation of it. And what we see here, each of these nodes is a different site. And each of these values next to those nodes is a different value for connectivity. And so what we have here in this network is we can see these sort of tips of these branches here would represent the headwaters, and they all sort of flow downstream and then connect down here to the main stem. And so in what is a typical pattern is that, the headwaters tend to be least connected from the other sites in the network because they're out on the very tips of the fingers, right? But then when the streams converge and you have this network come more down to the palm of your hand, the sites closer to the palm of the hand are more connected to each other. And so what these values here is basically just show the relative connectivity of each site within this network. And so this is just sort of showing that these headwaters are these sites that are just fundamentally least connected. And that's just because of network structure, right? Just because of the way the stream network is, the branches at the tip are more disconnected from the ones at the main stem. And so what we were sort of thinking about is, okay, so when a stream dries, when part of a stream dries, it's adding a layer of fragmentation. It's adding disconnection to this network, right? And so we were sort of wondering what might happen if a stream dries in the headwaters versus at a main stem. And so Albert, with collaborators, built this model. And so this model simulation results manipulated two factors. Let me get my laser pointer out here again. On this y-axis here, this is, in the model, they manipulated disturbance, or just sort of the amount of mortality that was induced by drying. And then here on the x-axis is the fragmentation that was induced by drying. And so the way to sort of read this is a simulation of the model. And so as we modeled this stream network, overlaid on these colors are alpha diversity, the number of invertebrate species that we might have at any given site. And so what we find here is the results in this model, when drying is very, very low, when the amount of fragmentation that is initiated by having a drying event in your stream, when that's low, regardless of how harsh that drying is on the community. So for this disturbance, as you go up here, like, we can make the disturbance with this mathematical model induce mortality in 100% of the time to 0% of the time, right? And so as mortality as a result of drying increases, even at low amounts of drying throughout the network, we still see relatively high levels of alpha diversity, right? And so what that means is that in the simulation, when there's not much drying in the network, even if the drying events are catastrophic and kills almost everything in the community, those populations can recover because we have dispersal from other sites that may not have dried. Or in some cases, we have taxa that are actually desiccation resistant and can tolerate drying and then grow back when they get re-wet. So next, what we show here is what happens on the other side, though, where when we have high amounts of drying throughout the network, only then do we see the effect of the disturbance magnitude begin to have an effect on the local richness here, right? And so what we're seeing here is, in this top part here, where we have very, very high amounts of drying-induced mortality and very high amounts of fragmentation, that is when we start to get significant impacts of drying, right? And so what this is sort of showing is that it's both the magnitude of the disturbance and the spatial orientation, the amount of fragmentation that's occurring from that disturbance. Only when those two interact do we get this negative effect on species richness at a given site. So that's the results of the model. And we can also play around with that model where we can actually look at where the drying is happening. We can force the models to induce the drying at the headwaters versus the main stem, or we can allow the headwaters to flow and then the main stem to dry. And so on this left, we have this upstream bias where we have only the drying is only occurring in these headwater upstream parts. And then it's free flowing in the main stem. Whereas in the downstream bias is then the drying events is happening in the main stem, but the headwaters are staying free flowing year round. And so with these graphs, again, we're sort of instead of manipulating magnitude of disturbance, we're manipulating position of that disturbance and looking at what the response is based on the site level connectivity throughout the network. And so what we find here is that when we have upstream based drying, it has the greatest effect on these isolated headwaters. And so with this green line, this is when we have fragmentation, this pink line is when we don't. And when we have upstream bias drying, we don't see much difference in these two lines, except at this very beginning part of the graph here, where the site connectivity is very, very low. So upstream bias drying is having its greatest effect on these isolated headwaters, whereas downstream bias effects here, what we see a difference between these two lines, where the fragmentation does have this significant impact across the entire community and the entire meta community, across all sites, across the network. And so what was really cool is what we could do is we could then test this model based on the data that we collected, because we had collected macroinvertebrates across all these sites. And so in this graph here, these solid lines show the modeled data, and then these dashed lines show the observed data. And what we see here is we see a decline. So we see the diversity is declining, but what we see is that our observed data are dashed lines. As a site dries from left to right, it's declining at a much greater rate than even our model predicts, right? And so this is, in the one sense, confirming that like this pattern that we're modeling is happening in nature, but we're still missing some things, right? There's still some things in our model here that we're missing out on, because we are still in nature losing losing species faster than we might anticipate when things are drying. So the next part of this talk here is to focus a little bit more on the ecosystem part, the food web part. And so with this part, we were wondering if drying reduces trophic diversity and food chain length. And so we were also thinking about questions about, does drying increase the importance of terrestrial energy sources? And then thinking about how these effects are consistent across climates. And so what I'm going to do is here show you some results from just the second part of this, the second and third, these two questions here, thinking about the importance of terrestrial energy sources in streams and how these effects vary across climates. So the first thing we sort of did as we were starting this is we kind of wanted to know at a broad scale how allochthony might vary in the first place. And so as we were planning the study, we decided to do a meta-analysis where we collected data from published articles, from stream consumers, aquatic invertebrates, and fish were what we focused on. But we're also interested in terrestrial consumers, the ones that live in the riparian zone, arthropods, and vertebrates. And so what I'm showing you here in these graphs these are the distribution of our study sites across the globe and through these different major climate zones. And you see we get, pretty good representation in North and South America and Europe, but a lot of Asia and Africa are lacking in representation. And then the bottom here is when we have our data from the different taxonomic groups. And we can see they're pretty well distributed across those straits that we're constrained in at first. So one of the things we wanted to look at first was, do aquatic and terrestrial consumers have equivalent allochthony? And so by allochthony, what we're talking about is the portion of the diet that comes from the other ecosystem. So for the aquatic species here, the fish and the macroinvertebrates, we're talking about the leaf litter and the falling macroinvertebrates that fall, if we go back to this diagram here. And then for the terrestrial predators, spiders and birds and things, what we're talking about for allochthony for them is when we have aquatic insects emerge and then they get consumed in the riparian zone. And so what we found was that the proportions of those allochthonous resources, regardless of the origin of the ecosystem, were largely equivalent. And this was a surprise to us. We thought that the aquatic species would be more reliant on terrestrial matter than terrestrial matter would be reliant on, then terrestrial organisms would be reliant on aquatic. And so that was sort of an interesting non-result there to not have in a finding. And then we also looked at how this might vary seasonally. And when we looked at this for aquatic macroinvertebrates up here in the top, across all these different seasons, temperate seasons, spring, summer, fall, winter, but then also tropical seasons for subtropical climates of dry and wet seasons. We found no variation in the amount of lochthonous diet, which is largely leaf litter that's consumed by aquatic invertebrates. But for fish, their lochthonous diet proportion is following aquatic insects. And so what we did find was some seasonal patterns there where the fish diets were much higher in the summer and the fall versus spring or winter compared to within the temperate climates, but then within the subtropical climates, no difference between wet and dry years. So that was an interesting result too. And then when we looked at climate zone, we found something a little bit different than we anticipated. What we thought we would have found for invertebrates was that we would have found that allochthonous diet contributions for invertebrates would be lower in arid ecosystems. But we in fact found that it was highest. And so we're not really quite sure what might be happening here at the time, although we had some ideas. And one of the things that we thought might be driving this is actually drying. And so we're going to circle back to drying here in a minute, but we're sort of thinking that drying might actually increase the reliance on terrestrial leaf matter in a stream, because when a stream is flowing and then dries, the primary productivity gets reset and suspended. So, and then for our climate zones with fish, we did not see anything different. So the next thing we looked at was how allochthony might vary by feeding group for aquatic invertebrates here up at the top and fish. And then we also did the same thing for the terrestrial predators that we looked at. Aquatic insects have different functional feeding groups based on, a lot of it's just sort of based on what we think they eat, based on habits and mouthpart morphology. So here we have collector, filter gatherers, predators, scraper grazers, and shredders. Shredders are the ones that eat shred leaves. And so we thought those would be highest, and those were, but the predators were also high as well. And then for fish, we have algivores, detritivores, invertivores, piscivores, and plantivores. There are some fish, particularly in tropical climates, that are actually adapted to feed on terrestrial nuts and fruits and things when floodplains flood. And so those were those fish. And those were largely reliant on terrestrial resources. Birds, it didn't really matter if they were an insectivore or an omnivore. They both really liked eating aquatic insects, apparently. And then for terrestrial arthropods, the web-weaving spiders consumed more aquatic insects than ground hunting in invertebrates, which included spiders, but also things like beetles. So that's sort of what we found. And so what we wanted to do was to think about how drying might affect food web structure in our system. And so to do this, we did a subset of our basins across the United States where we collected stable isotopes. And we had sort of 2 out in the arid climates in the West, 2 in the grassland, Great Plains systems in Oklahoma and Kansas, and then 2 that were sort of in our more mesic eastern forest systems in Mayfield and West Prong, Little Prong. And so what we found here was we had these 60 sites from 6 basins. And so what we did is we collected basal resources at each site, things like algae, bryophytes, and macrophytes. Those were sort of our allochthonous. And then our autochthonous was just leaf litter. And then we also collected our dominant macroinvertebrate taxa as well. And so first, I'm going to show you just sort of some of the raw isotope data. And so this was led by a PhD student, Kirsten Higgins. She's just writing this up now. We're going to submit this soon. So this is all really fresh and exciting to look at. But basically what we found here is this is our biplots of our basal resources. Our leaf litter, our allochthonous is in brown, our autochthonous is in green. And these are our different basins. And we do see, some differences. We see a lot more dispersion in the allochthonous resources than in the autochthonous versus the allochthonous, and then also that the autochthonous tend to sit somewhat higher up on the nitrogen. And so then what we do is then we take all of our invertebrates, we get carbon and nitrogen data from them too. And then this is just a biplot, just really just trying to show you that we have individual data for every invertebrate that we collected of carbon and nitrogen across these basins. And so what we do with all this data is we put this into Bayesian mixing models where we can then estimate the allochthonous and autochthonous contributions to diet. And so with these mixing models, we're a little bit limited in how we can build the model structure. For the first effect, we had basin or functional feeding group. Our second effect was an abiotic variable where we did either dry days, drainage area, wetted width, canopy cover, carbon nitrogen ratio of the leaf litter as kind of a measure of quality of that resource. And so we built models with different combinations of these to sort of figure out which one would influence the most. And so I mentioned drying earlier. And so again, we sort of thought drying might increase the reliance on terrestrial leaves within a stream food web, because when a stream dries, all the algae dies, and then it has to restart and it has to recolonize. Whereas actually the leaf litter stays in the drain and stays in the channel and actually might become more palatable because it still degrades. There's still microbes doing some work on conditioning those leaves so that when the water resumes, the leaves are there, but the algae is not. And then the variables that we're interested with watershed size are important because of the river continuum concept, which this is a well-known idea in stream ecology, where as a stream gets larger, as you go from a headwater down to a main stem, the importance of leaf litter or allochthonous contributions to the ecosystem become less important, right? And so basically, as you go from a small stream to a large stream, you should have more of an influence of algae or in-stream autochthonous production. So we ran all these models, and this was our best performing model where we had functional feeding group and drying. And so these are results of the mixed SIR model. Here we have our 4 feeding groups: collector filterer, collector gatherer, shredder, and herbivores. And the dietary proportion of either autochthonous resources in green or allochthonous resources in brown. And then for here, our continuous variable is proportion of dry days. So as the site's dry, we go from left to right. So what we're seeing here is that as we get more drying, we do see that these allochthonous resources increase in importance. And what is interesting is that this is true for all functional feeding groups. And if you know anything about invertebrates and functional feeding groups, one thing that is well known is that these are more sort of like general guidelines. And even if we classify shredders as being shredders, we know that they also will consume algae, for example. So the next model that was best performing was functional feeding group and area. And so this showed that trend that we sort of thought, where the drainage area, as you increase drainage area, you should get more wider stream, and we get more more autochthonous, less allochthonous contributions to diet. And then our third best performing model was basin and drainage area. And I wanted to show this one mainly just to sort of show some of the differences in basins. So for example, Sycamore Creek was that desert basin. This was very autochthonous driven. Pinnacles Creek is also very arid, and in California, that one was a little bit more mixed. And then we have King Creek, which is in Great Plains. This system was more allochthonous driven than any of them. And so these are the results of the model rankings. And what I'm sort of, though I'm presenting these models as like best and worst, we're still finishing up this analysis here. But what we get from this is what's called the DIC, which is similar to an AIC score. If you're familiar with that with model selection, it's just a Bayesian equivalent. And what we see here is we see these are all pretty close, right? So in terms of the models with a fixed effect, either functional feeding group or basin are important. But dry days are important, as our drainage area, and wetted width is also a different measure of stream size that we used. So really what the take home here is that what we found is that drying is as important, if not more so, than stream size for allochthony in streams across our systems here. So I'm going to shift gears a little bit to sort of talk about some other work that's going on in my lab that I'm excited to talk about. Some of it actually is neon related because it relates to another macrosystem project that we're starting up. But I just sort of wanted to briefly mention work ongoing in my lab where we're looking at stream biodiversity outcomes and how that influences stream restorations. And so this is an area of research that we've been focusing on here, because particularly around DC and Baltimore, within the Chesapeake Bay drainage area, there's a lot of stream restorations. And so what we try to do is, in a stream restoration, is we use mechanical equipment, bulldozers, excavators, to literally reconfigure the stream channel and then turn a stream that is something like this into something like this over time. One of the things that we find is that invertebrates typically fail to recover. And so here we have a figure just sort of showing, one of the reasons why benthic macroinvertebrates are really great to study is that they vary in how tolerant they are in pollution. And so some are very sensitive, and those are the ones that typically get knocked out in urban streams where there's all sorts of urbanization effects on the stream. And then we are left with sort of these more tolerant taxa. And the goal of the stream restoration is to make the habitat suitable so that we then can get these sensitive taxa back. And so this is called sort of the field of dreams hypothesis, where if we build it, then they will come. And so what we're sort of finding, though, is that they typically don't. And what we're wondering is, what if they can't get there? And so I have a student in my lab, Ibra, on a project funded by the Chesapeake Bay Trust, where we're basically testing this idea of doing some assisted migrations of stream invertebrates. We first did a short-term survival experiment that I'm going to present, and then we're doing this ongoing large-scale community translocation, which is, we're not going to have data yet for a while. But basically what we did here was we sort of picked 6 sites, and we put out these sort of habitubes, is sort of what we called them, sort of these tubes with a mesh and then rocks and leaves in them to sort of allow passive collection of invertebrates at donor and recipient sites, and then looked at whether or not the taxa survived the short-term transplant. And so these are our differences across sensitivity categories that the state of Maryland uses to determine their index of biological integrity scores. We see the decline in sensitive, moderately sensitive did not decline, and then a change in tolerant too. Looks like they might have increased slightly. And so we did see that, they weren't as abundant, but some of them did survive. And if we look at from our individual sites, what was interesting is that at 2 of these watersheds, Little Patuxent and the Potomac, those drainages we saw declines, but at the one and Loch Raven, we actually didn't see a difference. So all the sensitive taxa survived that short-term transplant at that site. And so, but we're getting variation in survival across sensitivity and basin. And so, but what is sort of really promising about this here is if you look at this more closely, we did this twice, once in the fall and once in the spring, and these green taxa are the sensitive taxa. And what this line here at 0 shows is that the abundance would be the same before and after the transplant. And so we do see a lot of sensitive taxa that are still abundant after the short-term transplant, both in the fall and in the spring. And so what we're seeing is that sensitive taxa can survive at these restored streams in the short term. And so what we're trying to do next is see if we can do these large-scale whole community transplants to try and repopulate. All right, so next I'm going to shift gears again. And last time I talked a little bit about environmental DNA because this relates to another macrosystems project we're working with NEON at starting this year, actually. So I want to talk a little bit about, just introduce the concept of environmental DNA. This is just the genetic material that is shed by organisms into their surroundings through skin, scales, feces, mucus, or what have you. We're all shedding environmental DNA right now. It's coming out of my mouth as I speak. And so typically what we do with invertebrates is we use traditional morphological identification techniques where we where we go into a stream, we collect a bunch of rocks, and then we take that sample and then sort and identify them. And typically what we do with a morphological ID is we typically we like this method because it's trusted. If we get raw abundance data, we know it works, but it's also takes a long time. It's very expensive. It requires a lot of training and it requires destructive sampling. eDNA is sort of this new method where we can think about maybe collecting the same type of data that's less expensive, it's faster. We might have better taxonomic resolution. It requires a more standardized training for molecular biological techniques that are taught in a lot of undergraduate molecular biology labs, for example. But the cons are that it's this newer method. It requires faith in DNA, and there are also some biases in terms of how we can interpret the abundance. And then the other thing is we don't really know much about how eDNA moves through streams. So one of the things we tried to do as part of this Stream Climbs project was we had 6 sites. So we at 6 of the basins across our project, we collected samples for metabarcoding, and we did some comparisons of our benthic samples versus the benthic eDNA metabarcoding, where basically we just take that sample and then blend it in a slurry and extract DNA from it. Or benthic and edge samples, where in addition to that benthic sample with a quadrat, we would also do a net kind of around the edge to sweep different habitats. And then we also did water eDNA sampling, where we used, where we filter water and then extract the DNA off that filter. And then we also just sort of compared the benthic eDNA metabarcoding with the water. And so what we found here was just looking at all taxa across all samples. This is just gamma diversity, like the number of raw taxa across all the different taxonomic levels with these different methods. The metabarcoding methods are both in purple and blue. The benthic, the morphological methods are in green and teal. And what we see a much higher abundance of taxa for the morphological methods at genus. But in metabarcoding methods, we see a lot more species identifications. And so we can look at this in some different ways. I'm just going to show that to you here. And there's one way is with taxa accumulation curves, where the amount of taxa that we get at a different sampling effort. At families, at the family level, we see more morphological versus the metabarcoding. Same thing for the genus. But then at the species level is when we start to see the metabarcoding get way more taxa. And another way to look at this is to look at our log response ratios. And so this is just where we're comparing the amount of, on a per sample basis, the amount of taxa collected in method 2 versus method 1. And so for the benthic morphological versus eDNA, this is a negative value, meaning the eDNA is getting less. So we're seeing less families with eDNA, less genera with eDNA, but again, more species with eDNA. When we look at the water, again, we're seeing less families and water-based eDNA, less genera and water-based eDNA, but more species. And then when we compare the two eDNA methods against themselves, we see more families with the benthic versus the water, same for the genera, but then no difference in the species. So then third way to look at this is just to look at linear regressions. And so here with the benthic versus the morphological, at the family level, we're seeing this line is less than a 1-to-1. So we're getting less families with benthic eDNA morphological, less generic. And then what's interesting here is we actually don't get anything relationship between species at all. And it's probably just because we're getting such few species identified with morphological methods. Same trend with the water. Again, we're getting less and less. And then here we're getting no trend. And then here on the right, we're sort of seeing something similar with the benthic, where the benthic is getting more species, more family than the water. And then we're seeing no relationship at the genera or species level for those two. So that's really interesting to think about in terms of the taxa that we're getting, but we're also interested in sort of the types of taxa that we're getting. And so to look at this, we can look at community compositions, where here we have species ordinations. And so what we're seeing with these different methods, it's the same coloring as before. The metabarcoding methods are over here in this ordination plot. Morphological are up here. And this is same for both family and genus. So we're getting different groups of taxa. And another way to look at this is with what's called an alluvial plot. And so here we have the four methods. These ribbons are sort of common taxa. It's present on the bottom, absent at the top. So you can kind of follow a ribbon to see a group of taxa and see which method it's contained in. So in here, this ribbon is like all four methods. This next one up is in the two benthic methods, missing in the metabarcoding water, but then back in the morphological edge, right? And so again, as we go down, from families to genus to species, we see most of the difference in the species. And so what's interesting about the species is that we're getting very few species overlap between the two methods, the morphological and the metabarcoding methods, but we're even getting significant differences in species overlap between the metabarcoding water and the metabarcoding benthic methods, which is interesting because it's the same methods that we're using in the lab. And so that sort of begs some questions about how eDNA moves in streams and how that might relate to what's being produced. So take homes from this is that we get different data from morphological metabarcoding, both biodiversity, taxonomic composition, but we're also getting different data from benthic eDNA and water eDNA, both in terms of richness and composition. And so what we're looking at in the future is like, are these differences meaningful in practice? Can we still use eDNA data the same way we might be able to in other ways? But also we're really interested in sort of looking at what causes these differences. How is eDNA moving in streams? And then how is eDNA produced? How does it vary by species, by organism type, by season? And so that's a project that we're just starting up this summer, where we're working with colleagues at University of North Texas, University of Alabama, where this is a project that we're calling Distance. This is another NSF-funded macrosystem project. And we're at NEON sites. This year, we've started already. We're doing eDNA sampling at all the NEON sites for water in conjunction with the biobouts so that we can compare seasonal differences in the composition in both the spring, summer, and the fall to get at some of that seasonal variation. And what we're going to start this summer is we're actually going to be doing eDNA injections at NEON weightable streams. And so we're really excited about that. We're starting up on the East Coast. We're starting that in May. And so with that, I will take questions.
Great. Thanks, Dan. I just want to remind everyone if we have a few minutes for questions, so put those in the Q&A, please. I was curious back to your network analysis project, what do you consider a dry stream? Is it totally dry? Is it disconnected? Does it have pools?
Yeah, that's a really good question. So for our study, we use these stick loggers, which are just for these stream temperature intermittency conductivity sensors. And so basically what they're doing is they're measuring water temperature, but then also conductivity. And so you can use the conductivity data to estimate if the water is flowing or not. And so we would put 5 of these out at our stream site, and we try and do different habitats, riffles, and pools. And so as we're analyzing this data, there are different ways in which we can classify that as drying. If one of those loggers is dry, then that means that there's some disconnection, right? It means that maybe it's have some disconnection pools, right? But then if all of those are dry, then it just means that there's no water, right? And so that's It's a really good question, and it's in practice a really hard thing to measure unless you're like at the stream. And so the way we did it was with sort of having multiple stick loggers in both riffles and pools, so we could sort of see how many of those five are dry and kind of make some judgment there.
Yeah, you would think something as straightforward as is it dry or not, would be easy to do, but we see the same thing at a lot of our sites, like sensors that are located pretty close to each other. One can be dry and then the other one's still in a pool or
Yeah, it's complicated for sure.
I still don't see any questions in the Q&A. If anybody wants to ask a question, we can also do that. If you just indicate in the chat that you want to ask a question, we can unmute you.
For your eDNA analysis, it looks like you're using a taxon-based approach. Is that right? Or are you?
Yeah, for that study, yes. But we are really sort of interested in these sort of taxonomy-free approaches, which maybe is what you've heard of. And so for people who don't know, basically what happens when you get all these DNA sequence data back from sequencing, and then you use bioinformatics to essentially separate them into what are called, it depends on how you do it, but most people do what are called ESVs, or ecological sequence variants, where you're sort of bioinformatically essentially sort of creating a mini phylogeny. And the ESVs are these sequences that are maybe like one or two sequence base pairs different from each other. And then the next step of bioinformatics, after you sort of build that out, is then if you're going to match those ESVs to taxa, then you have to match those sequences up to what exists in current databases. And there's some limitations to that approach, because it first requires the sequence that you're looking for the taxon to be in that database, and everything might not be sequenced. And so, but then there are some new approaches where they're trying to think of let's just maybe just because you do lose a lot of your reads if you're not able to match it up to a taxa. And so there's some new ideas about sort of using these taxonomy-free approaches. We're still trying to classify biodiversity, but just without our sort of typical approach of using taxonomy, and just sort of using the ESVs themselves, or OTUs are sometimes used too.
We have a comment from Karen. I was surprised to see no seasonal variation in macroinverts allochthony, as I'd expect seasonal variation in the amount of leaf litter that falls into the stream.
Yeah, that was really surprising too. We also hypothesized that we'd see a pulse in the fall. But, I think that, our sort of our best guess as to why that isn't is just because, when the carbon falls into the streams, a lot of times it's recalcitrant and it sticks around and it gets processed in different ways. And so I think, I think that's probably why we didn't see that, is because especially if you have species like oak, that carbon can stick around for more than a year. So
I guess we have time for one more from Bridget. Curious if you incorporated any remote sensing data in the first study about stream dryness, spatial patterns. I would imagine you might find some strong correlations with spectral water indices.
Yeah, so I will say there is a lot of data from that project that I did not present. There is a whole hydrological modeling part that used spatial data to parameterize those models, and we have a couple of really cool papers looking at how those drying patterns change. Because we could run those hydrological models under current conditions and climate change conditions, and so we did do that. I just did not have time to present everything that we did.
So we're at the top of the hour, so we're going to wrap things up. I want to thank Dan for presenting, and just a reminder, we have one more seminar coming up next month on May 12th. So thank you for joining us.
All right, thank you very much.